poster that I was presenting, we are basically showing how we make the electrical models for our column, so how all the ion channels are spread in the neurons that we are modeling, and how we try to automate the process of creating these electrical models so that we don't have to hand tune anything, that everything can be automated by our Blue Gene computer. The neurons are stimulated with this standardized protocol, which we call the E-code. They show a certain firing behavior, and you can see that certain cells give a very similar response, while some other cells fire in a different way. They start stuttering, they start firing irregularly. And so these electrical types is a way to classify the response of these neurons, so that we can also we don't create an electrical model for one specific cell, but we create an electrical model for a certain specific cell type, so that responds in a specific way to the protocol that we apply. So an electrical model is basically the way how we distribute the ion channels in the neuron, together with some extra mechanisms like calcium dynamics that we also put in the neuron morphologies. But it is the part of the model that let the neurons come alive. Basically, we first define our fitness function, so that shows how good our model is. Our model is parameterized, so we need to find the best parameter. So basically, every parameter describes like how ion channels are distributed in the dendrites and soma in the axon. And so our algorithm is a genetic algorithm, so it basically asks the evaluator to run our model with a certain set of parameters. That running means it applies the stimulus, the E-code protocol that they use in the experimental lab, to our electrical model. Our electrical model generates some voltage traces. These voltage traces are passed to our feature extraction library, which then calculates the action potential heights and all the different features in these traces. And so that creates values. So it tells, the resting potential of that neuron is minus 68 millivolts or something. So that value gets then compared with the actual experimental data, and we know in the experimental data there is like slight variations there in all these values. So we just calculate how many standard deviations from our experimental mean this value is away. And so this number of standard deviations, that's our fitness values. But we also have some idea what's happening in the dendrites of these neurons, of course. And for example, there's also backpropagating action potential in pyramidal cells, where action potentials move up from the soma into the dendrites. And so this is one feature that we also want to replicate. So our feature extraction library in our optimization algorithm also has ways of extracting or checking if there is actually an action potential going up in the dendrites. And so that's also something we've added to our optimization.